Click for IB Biology. Still with cell membranes, we're now going to look at a phenomena called the sodium-potassium pumps. There's a PDF of this presentation down in the description, linked in there, where you'll also find a link to 10 questions with answers, and in the description there's an AI prompt that you can copy and paste into a chatbot, and that will allow you to have individualized assessment in multiple choice style. To create the context of the sodium-potassium pump discussion, we could think about nerve cells, but I think it's important to state that all animal cells operate these sodium-potassium pumps in their cell membranes. And the consequence of this is that the inside of animal cells is always negatively charged, certainly in terms of millivolts, relative to the outside of the cell, which is positively charged. So this is not a phenomena which is unique to nerve cells. However, it does come up in the syllabus. It comes up within this context. So let me just explain that context to you. This is an attempt to draw a motor neuron, a motor nerve, and the direction of impulse travel is from the cell body here, travels along this axon to the end of the cell, which is called the synapse here. This part of the nerve is called the axon, and the nature of a nerve impulse is a wave of sodium ions flooding down the inside of the axon from the cell body towards the synapse. And so the direction of travel is this way, as indeed is shown on this diagram here. Now, although we're not going to go into the nature of a nerve impulse or how it moves in this video, what we are going to look at are the role of proteins embedded in the cell membrane and how they function as pumps in this process. To see the whole thing put together, you would need to address the section of the syllabus to do with nerve propagation, nerve transmission. So our sodium-potassium pumps are embedded into the wall all around the axon of the nerve cell like this. And that's what the subject of this video is about. Subsequent videos are on sodium gates, which are also in the nerve axon, and potassium gates, which are also in a video which follows that. So we've got 2 more videos on those 2 channel pumps as well. So we're going to address sodium-potassium pumping now. So in the cell, all around the cell membrane, there are many of these sodium-potassium pumps. And as I've already said, this creates a negative charge on the inside of the cell and a positive charge on the outside, relatively speaking. And the measurement, when measured in millivolts, the inside of an animal cell is approximately minus 60 millivolts, although there is some variation in different species. So however these pumps are working, the inside is negatively charged relative to the outside, and the difference is about minus 60 millivolts. And so to the mechanism itself, which is an ATP-driven membrane pump, an integral protein with 2 channels in it, one pumping sodium, which is here, and the other one pumping potassium, which is here. Notice that the pump, when active, is pumping out 3 sodium ions to 2 potassium ions. So if we think about each sodium ion has an associated negatively charged ion, such as a chloride ion, and the potassiums, which are out here, they would equally have an associated negative ion, like so. And so if we pump out 3 positive charges, but only return 2 positive charges. The net effect is to leave the inside overall negatively charged. And when measured with a millivolt meter, as I mentioned, this comes out to approximately 60 millivolts. There are all sorts of benefits of negative charges like this. Some of them involve the regulation of channels, the maintenance of ion gradients, and also in a way we're actually maintaining an energy store as we do so. That's the sodium-potassium pumps. What we're going to take a look at now are the sodium pumps, sodium gates, and then we'll follow that with a short video on the potassium gates, also involved in nerve propagation.